Hello everyone, welcome to my channel Online Biology Tutorials. We have already started with Chapter 6 Molecular Basis of Inheritance. One video has already been recorded on it. If you have not watched my video, please watch it. The link will be shared in the I button. Now, in the previous video, we have already talked about the different types of DNA. We basically need to concentrate upon the bDNA because that is very much asked in the board examination. And bDNA is a one that was given by Watson and Crick. So for grade 12 biology board examination, we will be talking specifically about the bDNA. We, whenever want to draw the double helical DNA, we need to be considerate that the two strands will be anti-parallel to each other. Means you can see here that the double helical DNA is shown and one strand is running from 5 to 3 prime, whereas the other is running from 3 to 5 prime. You can see here that minor and major grooves are also seen and they alternate with each other. Means first time it would be like minor, then major, then minor, then major. This way double helical DNA is there in the human body. Now very important part of this DNA is that we need to study the dimensions, right? And for studying the dimensions we need to open these DNA strands. So as I have opened, now it will be easy for you. Now the dimensions which are important to learn is the diameter, and this is the diameter of the double helical DNA, right? And this value comes out to be 20 angstrom, right? In NEET examination, they can also ask for the values in nanometer. So for that, you must remember that 10 angstrom is equals to 1 nanometer. That means 20 angstrom can be equal to 2 nanometer. So if they are asking you specifically in nanometers, please do make the conversions. Without it, don't mention. Now this one turn here, this is known as the pitch, and this length for about 34 angstrom, right? And there are 10 bases on each strand, so make sure whenever you are going to draw them, they should be 10 in number, and the bases on the other strand, they will be as per the base pairing rule. So here the first base is A, so here it is going to be T, then it would be A, then G, then it is C, then G, C, then A, T and A. And of course you know that whenever A binds to T, there are 2 hydrogen bonds in between and vice versa. So whenever T would also be attaching to A, there will be 2 hydrogen bonds. Whenever C attaches with G, there are always 3 hydrogen bonds and vice versa. Means whenever G will be attaching to C, there will be 3 hydrogen bonds. So you can see that 10 base pairs are present in one turn. So we will divide this length by 10 and we will get the value of 3.4 angstrom. Now what does this mean, 3.4 angstrom? It means that the distance between the base pairs, it is 3.4 angstrom. So pitch is important, distance between the base pairs is important, and diameter is also important. Now this was all regarding the bDNA. Now we need to know there was a scientist who was named as Chargaff. His full name was Erwin Chargaff, and he gave a rule that was known as Chargaff's rule. So what does this Chargaff's rule means? This is important. Numerical problems during the board examinations, they come based upon this rule. Now Chargaff said that the double helical DNA, it always follows a rule. And what is that rule? that the purines will always be equal to the pyrimidines. Now, in the first video, we have already discussed what are purines and pyrimidines. Just to recapitulate, let me tell you, purines are the double ring heterocyclic structures, and it includes adenine and guanine, right? And pyrimidines, if we talk about, then it includes cytosine and thymine. Now, it is very tough to write the complete name every time. That is why a common terminology is followed round the word, where adenine is abbreviated as capital A, guanine as capital G, cytosine as capital C, and thymine as capital T. Now here, if I want to explain, then I can explain it like the amount of A will always be equal to T, or if the value of T is given to you, then it will always be equal to A. Similarly, if I take the case of G, it will always be equal in concentration to C. Similarly, if the concentration of C is given to you, then it will be always equal to G. So this was Chargaff's rule. Now, basing upon it, numericals are asked. So let me take one numerical through which the concept will be clearer to you. Now, suppose in the question they give you that the concentration of A is 23% and find out the concentration of remaining bases. Now, how to proceed with it? You can first estimate. If A is given to you, that means the remaining three bases T, C and G concentration has to be found out. Okay, one thing is clear, because DNA is made up of 4 bases: adenine, thymine, cytosine and guanine. When A is given, that means we need to find out the remaining 3. So just now we have seen that the purine will always be equal to the pyrimidine, and as per the base pairing rule, A always binds to T. And as per the Chargaff's rule, the concentration given for A will always be equal to the T. That means if A is 23%, then T will also be 23%. Right? So we have found out that one purine is 23% and that is T, right? So if we add on A plus T, that means our value will become 23 plus 23 is equals to 46%. Now anything could be 100%, right? So what we will do, we will subtract this value which we have calculated, and that is 46, and the answer will come out to be 54. And that means one thing is sure now, cytosine plus guanine, they are equal to 54%. And one thing is also sure, that C will always be equal to G, because Chargaff proposed it. Right? So that means what we need to do, we need to simply divide this value by 2, and what it will give us, it will give us 27%. That means C is 27%, G is 27%. G is purine and C is pyrimidine. And purine is always equal to pyrimidine. As per the base pairing rule, C always binds to G and G always binds to C. So here A has come out to be 23% and T has also come out to be 23%. Now if you add on all the values together, what you will find out? You will get the value of 100%. This is just to cross check that we have done the addition right or not. So I will certainly recommend, if you will find out the values of each base in the end, do cross check them by adding them, because if any mistake in the addition would have been done, it can be traced in the early stage and it will give you full marks. So you need to prepare the numericals on Chargaff's rule also. I hope this is clearer to you. We will catch you in the next video. I hope you are sharing my videos, and if you haven't subscribed to my channel yet, please do so. Thank you. See you in the next video.